So, good evening, everybody. Just in 2 minutes, we'll start the class. So, we'll just wait for 1 2 minutes for others to join and then we'll start the class, okay? Okay, so let us start the class. So, today we'll talk about the lectures that are covered in week 8. And week 8 is about mechanisms of RNA decay and non coding RNAs. So, in the previous class we have talked a little bit in the last we have uh, talked a little bit about what is RNA decay and how RNA decay is dependent upon the 5 prime and 3 prime ends of the RNA. So this we have talked about little briefly in the class. So just if you I just wanted to give a quick overview of RNA decay and what we have talked about. So when I talk about RNA decay, this is your RNA. This is the 5 prime end of the RNA, and this is the 3 prime end of the RNA. RNA decay means that RNA is being attacked by some kinds of nuclease activity. Okay, it can be your exonuclease or endonuclease. Now, these nuclease activity, when it acts upon the RNA, so it starts degrading the RNA, and as the RNA gets degraded, what happens? That RNA is completely exhausted and RNA is completely lost. That is what RNA decay essentially means. Okay, so now the two major factors, the two major determinants that, just a minute. Okay, so the two major determinants that decides the stability of the RNA are its 5 prime and 3 prime ends. Okay, so what are the two major determinants that it determines the stability of an RNA? It is the 5 prime end of the RNA and the 3 prime end of the RNA. Why so? Because the 5 prime end has the 5 prime capping and the 3 prime end has a 3 prime poly A tailing. So what happens? Because of this 5 prime capping and 3 prime tailing means polyadenylation. Poly A's poly A's are attached, and here 7-methyl guanosine cap is attached at the 5 prime end. So because of this capping and tailing, the RNA, the extreme ends of the RNA are protected by any kind of exonuclease activity. What activity I'm talking about? Exonuclease activity. And since the RNA is protected from the exonuclease activity, the chances of RNA decay will be less. Also, we have studied in the previous class that the number of poly A's that are attached at the 3 prime end will decide the age of the RNA. Why so? Because if an RNA is a newly made RNA, then a lot of poly A's would be attached at the 3' end. As the RNA starts aging, what happens that these poly A sites tends to get degraded or removed because of the exonuclease activity, because of the exonucleases which continuously attacks the RNA. So when the RNA is aged, then number of poly A's would be less. So poly A are lesser in the case of an aged RNA, and there would be more poly A's in a newer RNA. So this means aged RNA is more prone to what? It is more prone to RNA decay, aged RNA, because there are less number of poly A sites, so the 3' end is getting even more susceptible to the attack of the exonucleases. Okay, so this is what we have in the end of our previous class. We have started to touch on this topic. So now we'll be talking about what are the RNA decay pathways. So we know that RNA decay happens, and gradually the RNA gets completely decays. RNA is completely decayed. There are different pathways that are taken for the decay of the RNA. When we talk about the decay of specifically mRNA, okay, we are talking about the mRNA decay. There are three major pathways. Okay, so when we talk about mRNA decay, there are three major pathways. What are those three major pathways? Those are deadenylation dependent mRNA decay, deadenylation independent mRNA decay, and endoribonuclease mediated mRNA decay. Deadenylation independent is the majority one. Okay. Now let us try to understand what is the meaning of this. Deadenylation dependent. First we'll talk about deadenylation dependent. What is the meaning of the term deadenylation? D means something is getting removed. Adenylation means the removal of adenyl group, that is A. So when adenine is getting removed, that is your deadenylation. So deadenylation dependent mRNA decay means shortening of the poly A tail by deadenylation enzymes. This step is reversible. That means that in the 3' end, just as we talked about just now, there are poly A sites. So deadenylation happens, and these poly A sites starts getting shortened, and the the A's are removed, and this causes this deadenylation is followed either by removal of 5' cap by the decapping machinery and 5' 3' decay, or 3' 5' decay. This means that deadenylation can trigger both 5' to 3' decay also, and 3' to 5' decay also. So whenever we talk about mRNA decay, decay can happen in three ways. There are three ways in which mRNA decay can happen. What are those three ways? The first way is, sorry, yeah. So the thing is, my mouse is not working today. I don't know why. Yeah. So first, it can be if this is your mRNA, okay, this is your 5' end, and this is your 3' end. So your decay can happen from this side. So it is from 5' to 3' decay. Okay. Decay can happen from this side. So it will be 3 prime to 5 prime decay. This is 5 prime to 3 prime decay. This is 3 prime to, or decay can happen if some endonuclease comes and it attacks on in between somewhere. So it would be attacked by the endonuclease. So what happens when we talk about deadenylation dependent RNA decay? Okay, I'm talking about deadenylation dependent RNA decay. So what happens here is that your because poly A tails are getting shortened from the 3 prime end, this can trigger either the removal of the 5 prime cap and eventually starting the 5 prime to 3 prime decay, or directly the 3 prime to 5 prime decay. So deadenylation can trigger both 5 prime to 3 prime decay also and 3 prime to 5 prime decay also. This is what it means. See here, shortening of the poly A tail followed by either removal of the 5 prime cap by the decapping machinery, or 5 prime to 3 prime decay, or 3 prime to 5 prime decay. This is your deadenylation dependent mRNA decay. We have deadenylation independent mRNA decay and endoribonuclease mediated decay also. So let us see. Okay, so this is the mechanism. First we'll see the mechanism and then I think we'll move to deadenylation independent. Fine. Now, briefly, we talked about the deadenylation dependent mRNA decay. What is the mechanism by which deadenylation dependent mRNA decay happens? So, first thing, which is the trigger point, is the shortening of the poly A tail. And shortening of the poly A tail happens with the help of deadenylation enzymes. This is important to remember. This is the name of those deadenylation enzymes. So, you see here, this is your mRNA. This is the 5 prime UTR. This is the 3 prime UTR. Okay. Hmm. So you see here, this is the 5 prime UTR and this is the 3 prime UTR. In the 5 prime UTR, what you have? You have the capping. 7-methyl guanosine cap is there. And in the 3 prime UTR, you have the poly A tail. So first step is the shortening of the poly A tail by the deadenylation enzymes. What are those deadenylation enzymes? They are known as CCR4 not or PARN. So this is that deadenylation enzyme. See, it comes and it has shortened the poly A tail. See, earlier it was 4, now it is only 2. Now what happens? With this shortening of the poly A tail, two things can happen. First is either 5 prime to 3 prime decay can occur, or 3 prime to 5 prime decay can occur. Anything can happen. Anything can be triggered by this enzyme. If 5 prime to 3 prime decay has to happen, then what will happen? This decay will only happen when the 7-methyl guanosine cap is removed. So this is known as decapping. So there is a decapping machinery that comes and it removes the 7-methyl guanosine cap. You see, DCP1, decapping machinery. This is the decapping machinery, DCP1. It comes along with DCP2. It takes the 7-methyl guanosine cap. So 7-methyl guanosine cap is now gone. Now it is very much prone. The 5 prime end has no protection. Now the exonuclease can come and eat it off. So that's what happens. This is the exonuclease. What is the name of this exonuclease? XRN1. It is again important. Not just exonuclease, exoribonuclease, because it is acting on RNA. So see here, after decapping, the 5 prime end is no longer protected and therefore 5 prime to 3 prime end exoribonuclease XRN1 degrades the mRNA completely. So remember that 5 prime to 3 prime ribonuclease is XRN1. Okay. So it causes 5 prime to 3 prime decay. Important. Now, this is about 5 prime to 3 prime decay. So, okay, one thing which is I just forgot to mention, that for the 5 prime to 3 prime decay to happen, what happens is that first LSM 1 to 7 complex, it gets attached at the 3 prime end, and then the decapping happens. So you have to understand this. First LSM1 uh, to 7 gets attached, and then your decapping machinery comes and takes away the 5 prime cap, and then your XRN1 comes and it removes the this thing. It cleaves and start decaying the RNA from the 5 prime. This is from the 5 prime end. And in the 3 prime end, what happens? If 3 prime and dependent 3 prime to 5 prime decay has to happen, then what happens? Exosomes comes. So it is very important here. Again, it is very important. So what happens? Exosomes come and bind at the 3 prime, and then they starts eating off the mRNA from the 3 prime end. So exosomes, they show their activity from the 3 prime end. So these two are important points here. Okay. So I hope this particular part, because this particular part is actually important. So that's why I'm giving a little bit more time for you to understand. There are several mechanisms that you will be dealing in this particular lecture. Okay. There are many mechanisms. Not all the mechanisms are equal in, I mean, for information per se, everything is important, but this is very much important with respect to your competitive exams point of view also. That's why I want to give more time to it. So I hope this particular deadenylation dependent pathway is clear to you. Okay. Now we'll see the next one, which is the deadenylation independent pathway, and then endoribonuclease mediated mRNA decay. So what are we learning here? We are learning about the different pathways through which mRNA is decayed. So there are three pathways through which mRNA is decayed. First is deadenylation dependent mRNA decay. Second is deadenylation independent mRNA decay. And the third one is endoribonuclease mediated mRNA decay. Okay, so there are these three pathways. So now we'll study the deadenylation independent mRNA decay. So deadenylation independent mRNA decay means what? Just a minute. I'm just switching the screens because I want to see whether the recording is happening or not. Because for, I think for the last to last class, I set the record button on and I don't know what happened. It didn't record. So now I had to record the whole session again for you guys. So I just want to make sure this doesn't happen this time. Anyways, so now we'll see the deadenylation independent mRNA decay. So here, since it is a deadenylation independent pathway, that means there is no need of any enzymes or any mechanisms that will remove the poly A tail from the 3' end. Okay, there is no need of removing the poly A tail from the 3'. So what happens here? It requires direct removal of the 7-methyl guanosine cap from the 5' end of the mRNA. There is no need of this and that to attach. So what happens? First, this RPS 28B, it's a protein. It comes and it binds at the 3' end. And then what happens? Sequentially, EDC3 comes and binds. So these two proteins goes and binds at the 3' end. And this facilitates the entry of the decapping machinery. So you remember, decapping machinery is same for deadenylation dependent as well as deadenylation independent pathway. The end thing is that decapping machinery should come at the 5' end. Now, whether this decapping machinery is coming because of the removal of the poly A tails, or this decapping machinery is coming because some other proteins are coming and binding at the 3' site, that is a different story. The important point is that this decapping machinery should come and it should bind at the 5' end. That is what is important. So in the previous case, the coming of the decapping machinery was induced because the poly A tails were removed and some other proteins eventually came and sit at 3' end. But here the decapping machinery is coming because of these proteins are coming and sitting at the 3' end. But in either cases, the decapping machinery remains the same. So it will come and it will bind at the 3' end and it will facilitate this XRN1 to come and chop off the mRNA from the 5' end. Okay. So here again you see decapping machinery remains the same. 5' to 3' exonuclease XRN1 also remains the same. So these two things are the same here. Okay. Third is endoribonuclease mediated mRNA decay. So we have studied deadenylation dependent, deadenylation independent, and now we'll see endoribonuclease mediated. So basically, this is your mRNA. Okay. This is your mRNA. This is the 5' end of the mRNA, and this is the 3' end of the mRNA. Decay means chopping off the mRNA. So mRNA can be chopped off from this side. It can be chopped off from this side. It can also be chopped off somewhere in the between. For somewhere in between to be chopped off, we need some kind of ribonuclease which can act in the interior, in the inside. And those ribonucleases are known as endoribonucleases. You must have studied about two kinds of nucleases. The first one are the exonuclease. Second one are the endonucleases. Correct? So exonuclease which acts at the exterior ends, 5' and 3', and endonucleases are the nucleus which act somewhere in between. So this is acting somewhere in between. So what happens? Cleavage by an endoribonuclease produces two RNA fragments that are susceptible to decay by exoribonuclease. See, things again, it is happening in the same way. This is my mRNA. Just a minute. Yeah. See. Hmm. See, again, it is happening the same thing. This is my mRNA. This is the 5' end. This is the 3' end. Some exonuclease came and it cuts at the between. Here it is cap. Here it is poly A tail. So now what happens? I have now two RNA fragments. One has a 5' cap. This becomes a 3' end. This becomes the 5' end, and at the 3' end, I have the poly A tail. So because some endoribonuclease came and attacked at in between here. Okay. Some endoribonuclease came and it attacked here in between. It has produced two mRNAs from this mRNA. Now we know that if we have any open ends, then exonuclease can come and easily bind to it and it can easily chop off the mRNA. Here, because of this, what has happened? It has produced here one open end at the 3' end and it has produced another open end at the 5' end. So two open ends have been produced. Okay. Two open ends have been produced. So because of these open ends that have been produced, here 3' to 5' decay can happen, and here 5' to 3' decay will happen. Okay. So how 3' to 5' decay can happen? 3 prime to 5 prime decay can happen because of the activity of what we have just now studied, exonucleases. So exonuclease will come, it will start chopping off from the 3 prime end. 5 prime to 3 prime decay can happen with the help of what we have just now studied, XRN1. XRN1 will come and it will start chopping off from the 5 prime. So let us see whether what we have discussed makes sense and this is what they are talking about. Yes. See here. See. They have cut it. Endonucleases came. They have cut the mRNA into two parts. You can see here. Now you have a 5 prime protected end, but this newly made 3 prime end is open. Here you have the 5 uh, uh, 3 prime end which has the poly A tail, but newly made 5 prime end is broken. So here at the 3 prime end, exonuclease will come, it will start chopping off the mRNA. Here at the 5 prime end, XRN1 will come and start chopping off the mRNA. So in this way, endoribonuclease mediated mRNA decay happens. Now what is there? Endoribonucleases are highly specific and highly regulated. That means, what does it mean? It means that it can come and it will bind at a particular site and then only it will do its action. It cannot just randomly come and bind to any place and it can start its action. No. They are highly specific and they will come and they will bind at a particular site and then only they will do the action. Okay. So I hope this particular, the different pathways that are required for mRNA decay are clear to you. Okay. So now this was in lecture 1. So in lecture 1 what we have essentially studied, in lecture 1 we have studied about the different pathways that are required for mRNA decay. What are the different pathways? Deadenylation dependent, deadenylation independent, and endoribonuclease dependent pathway. Now we come to study about the p bodies. What are p bodies? So p bodies are short form and the full form is processing bodies. So p body stands processing bodies. So what are these p bodies? P bodies are cytoplasmic ribonucleoprotein, that is RNP granules, which are primarily composed of translationally repressed mRNAs and proteins which are related to mRNA decay, suggesting their roles in post-translational modification. So basically, all those mRNAs which are going to be translationally repressed. Repressed means that they are damaged and they are now these RNAs are marked for degradation. So those RNA which are translationally repressed, so along with some proteins, they come and they make P bodies, which are processing bodies. They are marked for RNA decay. So always remember that P bodies are composed of translationally repressed mRNAs and proteins, which are related to mRNA decay. So what happens? You see this image. Just a minute. Okay. Yeah. So you see this image. So what happens in a normal cell? Normal cell has both LSM1 and XRN1. What is LSM1 and XRN1? Can anybody tell me? We have just studied at least XRN1 you should be able to tell. What is XRN1? XRN1 is the exoribonuclease. Na? This is the exoribonuclease that works in the direction of 5 prime to 3 prime. You see here, you should remember this because then only these p bodies will make sense. See what happens. We have studied here that LSM1, for 5 prime to 3 prime decay to take place, what happens? First, LSM1 binds at the 3 prime end, and then it recruits several decapping machinery, and decapping machinery removes the 7-methyl guanosine cap, and ultimately SXRN1 comes and it decays. This means that in one mRNA, both LSM1 and XRN1 are present. Could you understand? Whichever mRNA is marked for decay, try to understand my statement. Whichever mRNA is marked for decay, that mRNA should have both XRN1 and LSM1 to 7 both. Both should be present on that particular mRNA which is marked for decay. And these are what? These are proteins. That means P bodies are what? These are translationally repressed mRNAs. Those mRNAs which cannot undergo translation, which cannot complete their fate. So those mRNAs should be decayed. So P bodies have those mRNAs which have to be decayed plus some proteins which helps in its degradation. Okay. So here they say a normal cell has both LSM1 and XRN1, but they do not come condition. Now what happens? Under stress condition, RNA is damaged and it is marked for degradation. So LSM1 and XRN1 both come together. You see, normal conditions, there are not much yellow colored cells. Yellow means both LSM1 and XRN1 are together. Okay. LSM1 means that both yellow color means that both LSM1 and XRN1 are together here. So when both LSM1 and XRN1 are together, this means that that particular mRNA is marked for degradation. That means a P body is present there. So if you want to see under stress, if you want to see whether a cell has P bodies or not, what you do is take normal condition mRNA, stress condition mRNA, I mean cells, and then you, then you do the fluorescent staining with both LSM1 and XRN1. Then if it gives you yellow color, this means that those particular mRNAs have formed the P bodies. Okay, so I hope this is clear to you. This is what P bodies essentially means. It is important for us to understand what is the meaning of P bodies. Okay, now let us try to understand the structure of exosomes. So what are exosomes? Exosomes are nothing but the majority of the 3 prime to 5 prime exoribonuclease which are present in the eukaryotes. Okay. So exosomes are the 3 prime to 5 prime exoribonuclease, and they are present both in nucleus as well as in the cytoplasm. And what are the function of this exosome? This exosomes helps in the degradation of the mRNAs. It also degrades aberrant non-coding RNA, means those mRNAs which are non-functional. So those mRNAs are degraded by the exosomes. So exosomes, what it does, it degrades the non-functional mRNAs. Okay. Then processing of precursor of non-coding RNA to functional coding RNA, that is okay. So RNA degradation complex which targets RNA from 3 prime to 5 prime end. This is what is important. If anybody asks you about exosomes, this you have to tell. We were talking about 3 prime to 5 prime exonucleases. So these exosomes are nothing but those 3 prime to 5 prime exonucleases. So these are RNA degradation complex which targets the RNA from the 3 prime end to the 5 prime. Okay. Then here it is in brief, this is the exosome structure. So there are basically 9 different proteins, and these proteins form a ring shaped structure. Okay, so these 9 proteins which are present in exosomes, they form a ring shaped structure, and it consists of these proteins: R RP41, RRP42, RRP43, 45. Just a minute. Hmm. Hmm. And so on. And the ring structure is not stable. It requires some additional factors. It forms a cap. And this is all just the cap contains some RNA binding domains and facilitate the interaction of the exosome with its RNA substrate. So this exosomes has actually, it has actually to go and bind to the RNA substrate. So this cap actually helps in the binding of the exosome with the RNA. So this is just in brief about how, the structure of exosome, but essentially what you should remember is that this exosome basically degrades the RNA from the 3' to 5' end. So these are nothing but RNA degradation complex which targets the RNA from 3' to the 5' end. This is what exosomes essentially means. So now, how exosome helps in the regulation? So basically exosomes is involved both in processing of the mRNA, and it is involved in degradation of the mRNA, and it functions both in nucleus and cytoplasm. So you should always remember that these exosomes, when I talk about exosomes, they are involved both in processing of mRNAs, and they are also involved in the degradation of mRNAs. Okay. So a number of different proteins and protein complexes have evolved that guides exosome to distinct classes of substrate RNA. Okay. The best two studied complexes are TRAMP and SK1, SKY. So these are the two well studied examples of exosomes and its regulation. So let us study about this TRAMP, TRAMP. So this TRAMP complex consists of AIR, AR1P, AR2P, TRF4P, TRF5P, MTR4P. So these all, what happens that what these are the complexes, and basically what happens that these complexes helps in guiding the exosome towards its RNA. So if this is your exosome, it has several complexes, right? Now this exosome has to be guided towards the RNA. So these different individual substrates which are present, they guide the exosome towards the RNA. Okay, so this is what TRAMP complex do. And SKY complex, what happens? SKY complex also has different substructures, subunit structures, and especially SKY 7P, because SKY 7P, what it does is that it recruits the exosome to abnormal translational events and thus degrade the mRNA to prevent the synthesis of aberrant and potentially harmful protein products. So that means that this SKY1 complex, SKY 7P, it recruits the exosomes to the abnormal translational events. So these are nothing but the two different exosomes categories that are there. Okay. Now we come to the next thing, which is mRNA surveillance. So what is the meaning of surveillance? Can anybody tell me first? What is the meaning of surveillance? Hmm? What is the meaning of surveillance? Means monitoring something. When we say that it is under surveillance, something is under surveillance, that means we are completely monitoring or completely keeping an eye on something. Okay, so like we say that uh, 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 that particular CCTVs. in very general language, if I want to explain you, then CCTVs. So what are CCTV? They are actually uh, a method or a way of keeping an eye on what is happening there. So it's like a video form of a surveillance. So when I talk about mRNA surveillance, mRNA surveillance means that we are keeping a quality check on the mRNA molecules that are made. Okay, so keeping an eye and keeping a quality check, and quality check, when I talk about, then I talk about that whether the mRNA is good or not, or it is of high fidelity or not. So mRNA surveillance means keeping a quality check on the mRNA. And how we'll keep the quality check? Primarily by detecting and degrading the aberrant mRNAs. Okay, so whatever aberrant mRNAs are there, means non-functional mRNAs are there. So first, to detect and then degrade the aberrant mRNAs or slow mRNA or non-functional mRNA. Okay, so this is what mRNA surveillance means. So what it is saying, that when I talk about mRNA surveillance in the nucleus, so basically we are keeping a quality check. We are keeping an eye on the improperly or aberrant mRNAs, and how we are keeping an eye on the improper aberrant mRNAs by degrading the bad ones. That is what we can do, degrading the bad RNAs. So this degrade, because here we are talking about RNA decay. So everything has to do something with the degradation of the bad RNAs. So mRNA surveillance is just a fancy word you can say given to it, where degradation of aberrant or improperly processed RNA happens in the nucleus. So now how to say that a particular mRNA is a bad mRNA or a non-functional mRNA. So basically, mRNA can be a good mRNA or a aberrant mRNA, and this can be detected on the basis of certain categories. And what are those categories? First, if it has improper five prime capping or a five prime three prime tailing. If the 5 prime cap is not proper and 3 prime cap is not proper, then we can say it is a good aberrant mRNA. Second, which is very important, is that it has a malformed mRNPs. That means ribonucleoprotein complexes which are formed that are also not functional. Then also we can say that those RNAs are aberrant mRNAs. Then it can have either fast polyadenylation or slow polyadenylation. So what is the meaning of fast and slow? The polyadenylation event, if it happens fast, then it's a good RNA. No need of this mRNA surveillance. But if the polyadenylation which is happening is very slow, then we can say that this mRNA is not good. And it can be marked for mRNA surveillance or mRNA degradation. So this is again some category. There are some categories. What is first category? First is either it can have a bad 3 prime end or it can have a bad 5 prime end. It can have a malformed mRNPs or it can have a slow polyadenylation. So whatever these things happens, so what happens, that this tramp, it trims the poly A tails of RNAs, and then what happens, exosomes comes and it starts the degradation of that particular marked mRNA. Okay, so this is what you have to understand more. So I hope this thing is clear to you. Okay, now again important point to be discussed is mRNA surveillance in the cytosol, regulated mRNA decay in the cytosol. So what we have studied here, these are the mRNA decay that were happening in the nucleus. Now we'll see ki in the cytosol how mRNA decay can happen. Okay, again important point, mRNA decay can happen in the nucleus, can happen in the cytosol. We'll see how mRNA decay can happen in the nucleus now. Okay, so interesting. Let us try to find out. Sorry. Hmm. So have you guys studied about mutations? Generally, I'm not talking about right now. I'm not talking about the mRNA decay. I mean, general talking about mutations that happens in the body. Have you studied about mutations? Hmm? I think you all must have studied a little bit about mutations. So mutations can be of various types. Okay. It can be a missense mutation. It can be a nonsense mutation. Have you studied about these two mutations? The first category is missense mutation, and the second one is nonsense mutation. So, what happens in missense mutation if suppose it is ATG or AUG, whatever it is. Suppose this AUG, it codes for methionine. Okay. This particular AUG, it codes for methionine. And because of missense, now this G is replaced by, let's say, C. Just for example per se, I'm saying, okay. So now this AUG has become AUC. So now the sense of this codon has been changed. Earlier, this codon used to code for methionine. Now, let us say it codes for tryptophan. I'm just saying, I'm not saying that AUC codes for tryptophan. I'm just giving you an example, arbitrary example. Okay. Don't say that, ma'am, how you can say AUC does not code for tryptophan. I'm just giving an example. So now this AUC codes for tryptophan. So the sense has been missed now. The sense has been changed now. So now this particular mutation is called as a sorry, is called as a missense mutation, because whatever sense this mutation earlier used to make, that sense has been changed now. It doesn't make the same sense as it originally has made. So this is known as a missense mutation. Then I come to nonsense mutation. Nonsense, what, we say this is nonsense, it doesn't make, which doesn't make any sense. So nonsense mutation means uh here. So nonsense mutation means that now suppose there was a codon AUA, and suppose it codes for any alanine, let's say. Now it has been changed in such a way that it becomes UUA. Now what does UUA stands for? Can anybody tell me what does UUA stands for? UUA, UAG, UGA. UUA stands for a stop codon. It stands for a stop codon. So what has happened here? That a particular codon has been mutated in such a way that now it became a stop codon. So when it became a stop codon, suppose this is the mRNA and here there was AUA. Now this AUA changed and it become UUA. So what will happen now, that since it became a stop codon, so now the machinery will get the idea, will get the instruction that stop codon has come and we have to stop the protein synthesis. We have to stop the translational machinery. So your protein synthesis will start here itself. It will stop, which means that this particular area which has to be synthesized will not be synthesized. So the protein which is formed is an incorrect protein because it is not completely formed. And since it is an incorrect protein, it will not show the proper folding also. So these are the two types of mutations which are important to understand. Okay, the first one is the missense mutation, where the sense of the codon has been changed. Earlier, that particular codon used to code for some amino acid, let's say methionine. Because of this missense mutation, now that codon states uh, uh, codes for some other amino acid, let's say tryptophan. So that is your missense mutation. And what is nonsense mutation? Nonsense mutation means that earlier that codon used to code for some AVs, any amino acid, but now it has been changed in such a way that now it codes for a stop codon. And since it codes for a stop codon, the protein, the mRNA synthesis will stop there only, and protein will be not formed correctly. Okay, so I hope this is clear to everybody. Now, with this background knowledge, let us try to understand the mRNA decay that happens in the cytosol. Why I was trying to explain you this and why I gave so much of time on this is because it is important for you to understand the concept at this stage. Okay, why? Because see, after that all are these pathways only. Okay, they not much to explain the pathways, and then we have the questions. Yeah, this is the main thing that I wanted to explain you here, because um in 6 7 lectures there are some important points that are your concept building points that will help you to understand the concept. And if your concept is clear, if your base is clear, then anything which is related to that concept, anything which is related to that particular base, it is easier for you to understand. If you know what is the meaning of nonsense mutation and missense mutation and the different kinds of mutation, you can very well and easily understand any terminology or any phenomena which attaches the nonsense and missense part. Now here comes the nonsense mRNA decay. So nonsense mRNA decay means it happens in such a way that if nonsense mutation has occurred in some mRNA, that mRNA is marked for nonsense decay. Let us try to understand in this way. If nonsense mutation has occurred in any mRNA, then that mRNA is marked and targeted for nonsense mediated mRNA decay. See here, target mRNA that contains premature termination codon. So how premature termination codon can come? Premature termination codon can come because of some problem in the synthesis machinery where the normal mRNA is mutated and it became the premature mRNA. So that is your nonsense mRNA decay. Non-stop mRNA decay means now the decay is not stopping only, non-stop, continuously decay is happening. So that is your non-stop mRNA decay. So it targets those mRNAs that do not contain a stop codon. There is no stop codon only at all. So continuously it is going on. Continuous mRNA synthesis is happening. That is your non-stop mRNA decay. Then you have no-go mRNA decay. What is the meaning of no-go mRNA decay? It targets those mRNA that form stable secondary structures which inhibits translation. So, means suppose this is your mRNA. Okay. Now, this mRNA forms a structure like this. So, now this is what? This is your hindrance. Okay. This secondary structure is nothing but a hindrance. And because of this hindrance, the translation cannot go after this. It cannot pass this particular point. Okay, so this is a no go mRNA decay. That mRNA cannot go after this secondary structure. So this kind of decay is known as no-go mRNA decay. So three kinds of decay happens in the cytosol: nonsense mediated mRNA decay, non-stop mRNA degradation, and no-go mRNA decay. So I hope this is clear. Let us try to understand it in a bit detail. Okay, let us see nonsense mediated mRNA decay. So nonsense means any kind of stop codon has come. Okay. So let us try to see. See what happens, that this is a stop codon. It's a premature stop codon. Okay. Now this premature stop codon has ERF3. Now ERF3, it looks for poly A binding proteins. If it finds poly A uh, uh, binding proteins, if it finds, then what happens? Proper termination can happen and mRNA can persist. But if by chance this ERF3 does not find the PAB, instead it finds the UPF1, then what happens? Then that mRNA will be marked for degradation. And these two events are mutually exclusive events. So when I say mutually exclusive events, that means anything can happen, either this or this. It all depends on the fate and the luck. What can happen? Either it can go for this or it can go for it. If it finds poly A binding protein, then proper termination can happen and mRNA can persist. If it finds UPF1, then it is marked for nonsense mediated decay and mRNA is degraded. So it all depends on the luck whether it will find PABP or UPF1. This is your nonsense mediated mRNA decay. Then I come to lecture 5. You see, this is just like a major more explanation of it. See, this is the stop codon splicing and other processing happens, and how this. So this is just like even more detailed diagram that you can go and just get a read through of it. I have explained you the major concepts. Okay. Again, when we talk about the decay in the cytosol, see, we have already studied that what has to come. If it's a 3' end, always remember whether it is a cytosol, whether it is a nucleus, 3' end pe what has to be attached? Exosomes has to be attached. 5' end pe what has to be attached? XRN1 is attached. If you remember these two things, it becomes very easy for you to understand. Your 7-methyl guanosine cap has to be removed. How it will be removed? With the help of some decapping machinery. So decapping machinery, DCP. DCP stands for decapping machinery. So these are some general ideas that you should remember. Okay. Now next. Just a minute. Yeah. Then we come to non-stop decay. So what happens? That there are two cases that can happen when we talk about a non-stop decay. What is the first case? The first case is stop codon is not there, or stop codons are there but ribosome is not able to stop it. So basically in non-stop decay, what happens? Then there is continuous translation happens. There is no stop to translation. Okay. This is your non-stop decay. Non-stop it is getting translated. So it can happen because of two ways. The first is that stop codon is not there, or stop codon is present but ribosome is unable to stop the stop codon. So these are the two ways in which non-stop decay can happen. So ribosome translates. What happens? See here. This is the poly A binding protein which binds at the poly A sites. Then ribosome translates to the poly A tail and it stops. What happens? When Sky1 comes and it binds to the ribosome. What happens if Sky1 comes and bind to the ribosome? Then this coming of the Sky1, it recruits the exosomes and it starts decaying. Or what happens if Ski1 is not there? Then there is a loss of poly A binding protein and which allows the decapping. So if Ski1 is there, see, if if Ski7 is present, then you will have 3 prime to 5 prime decay. If Ski7 is not present, then you will have 5 prime to 3 prime decay. So Ski7 decides whether it has to go for a 3 prime to 5 prime decay or a 5 prime to 3 prime decay. Is it clear to everybody? I hope that this becomes clear to you. Okay. Now let us study. Sorry. Yeah. Mm-mm. Now we come to no-go decay. Why? See, ribosome cannot go anywhere. It gets stalled in the middle. It gets stopped in the middle. And why it gets stopped in the middle? Because some kind of secondary structure has come and some kind of secondary structure has formed in between the mRNA. And because of that secondary structure that has formed in between the mRNA, now ribosome is unable to come and stop in between. Okay, this is what no-go decay means. So you see here, hmm, sorry. Yeah. So this is a some RNA structure has formed in between. And because of this RNA structure, the translation is stalled. And when the translation is stalled, some machinery comes and there is an endonucleatic cleavage. And when there is an endonucleate cleavage, then this mRNA forms two different segments. Here, this becomes the 5 prime end. This becomes a 3 prime end. Here, this becomes the 5 prime end. This becomes the 3 prime end. So this 5 prime end has cap, but 3 prime end is empty. So the exosomes will come. It will chop the mRNA from the 3 prime end. In this particular RNA part, the 3 prime end has the poly A tail, but the 5 prime end becomes empty. So XRN will come and it will bind to the 5 prime end. It will chop the 5 prime end, mRNA from the 5 prime end part. Okay. So this is how this decay happens. I hope this is clear to you. So two things again I am repeating. If you want 5 prime to 3 prime decay, then XRN1 has to come. If you want 3 prime to 5 prime decay, then exosomes will come. Okay, fine. Then comes auto-regulation of RNA. So basically, what happens? Sometimes to prevent the decay of the RNA, some kinds of auto-regulation events also takes place. Okay, so what happens? Here I will not be explaining you much because it is just like different examples that are given for the auto regulations. You just have to go through them. That auto regulation, there are different examples. First is auto regulation of NAB2 poly A binding protein. Second is degradation of meiotic transcripts that happens. This is also an example of auto regulation. Then exosomes and gene expression, here also you can see the example of auto regulation. So these are all the various examples of how auto-regulation happens. No need of going in detail in that much. Now we'll see. Now from here we come to the other important kind of RNA, which is the non-coding RNAs. So there are two kinds of RNAs. Okay, you must be knowing that there are two different kinds of RNAs that are present. The first is the coding RNA, and the second one are the non-coding. So what are the coding RNAs? Coding RNAs are the RNAs that codes for a particular amino acid. So which RNA type codes for a particular amino acid? Can anybody tell? The RNA that codes for a particular amino acids are the mRNAs. mRNA only codes for a particular amino acid. Rest all, they help in translation process, but they do not code for a particular amino acid. Okay, could you understand the difference? Rest other type, they code for a particular amino acid. No, they just help in the translation process. So whether we talk about the rRNA or we talk about the tRNA, all these RNA, they code, they just help in the translation process, but they do not code. Coding part is done only by mRNA. And there are other non-coding RNA also that are present. And what are those non-coding RNAs? Let us see. Those are snRNA, guide RNA, snORNA, and many different types of RNAs. Okay, just a minute. Wait a minute. Okay, fine. So if you have any problem, you can raise your hand from here and you can ask. Okay. Now, non-coding RNAs, what is the definition of a non-coding RNA? A non-coding RNA is a functional RNA molecule. Now, remember this term, functional, because earlier people, scientists or researchers used to think that non-coding RNAs are not functional. But now we know that even non-coding RNAs are functional. So, a non-coding RNA is a functional RNA molecule that is not translated in a protein. The only difference is that that particular RNA molecule is not translated into a protein. Rest everything is same. Now, even non-coding RNAs can be of two types. The first type is the housekeeping non-coding RNA, and the second one are the regulatory non-coding RNAs. Housekeeping are housekeeping. I don't know whether you know or not this terminology, whether you know or not the terminology which we call as housekeeping genes. What is the meaning of housekeeping genes? Housekeeping genes are those genes which are continuously expressed in the body, okay, and which are required for the normal functioning of the body. Those genes are known as housekeeping genes. So housekeeping non-coding RNAs again are what kind of RNAs? Which are constitutively, means constitutively expressed, and are required for normal function and viability of the cell. Okay, constantly these housekeeping RNAs are expressed and they are required for the normal functioning of the cells. And other are the regulatory non-coding RNAs. They are expressed only in certain stages of organism development. They are not developed constitutively. They are only developed expressed in certain stages or in response to some external stimulus. Okay, so two categories. First is housekeeping non-coding RNA, and second one are the regulatory non-coding RNAs. Housekeeping non-coding RNAs are required for the normal functioning and viability of the cells, and regulatory non-coding RNAs are required at certain stages, and they are expressed in response to some stimulus. Now, regulatory non-coding RNA can affect the expression of other genes at the level of transcription or translation. This is also important. This point is also important. Okay. This point states that regulatory non-coding RNA, it can also affect the expression of other genes, and it can affect the expression of other genes at the level of transcription or at the level of translation, both. Okay, there. Now we'll come to the question. So now, okay, this is fine. We have completed the 6 lectures course. I mean, 6 lectures, whatever there, and now we'll do the question. So, as what is the way we do the question is that I'll give you the question. I'll give you 2 to 3 minutes to answer the question, and then I will switch the screen, and then we'll discuss the answer. Okay, fine. So the first question is, exportin T function depends on A, RNA quantity inside the nucleus, B, RAN GTP, C, RAN GAP, and the last option D is exportin 5. So exportin T ka function is dependent on what? quantity, RNA quantity, RAN GTP, RAN gap, or exporting 5. So right now it is 8 8. I will switch the screen at 8 10. Okay. You have 2 minutes to answer this question. Think and try to answer. One more minute left. Okay, now I am switching the screen. Okay. Hmm. So, whether you have answered something or not. So, if you know the answer, you can just type the answer in the message box, okay? So, nobody has typed anything. So, couldn't understand this? This was something which we have dealt in previous class. So, okay, the correct answer is RAN GTP. Okay, so how it is RAN? Okay, how it is RAN GTP? Anybody can tell me? No? Okay, I'll tell you. See, what are exportins? First of all, you should know that what are exportins. So, exportins are transport receptors. Okay, when we talk about exportins, these are transport receptors that mediate the nuclear export of specific cargo proteins, which includes your RNA and proteins all. Okay, this process is RAN GTP mediated. When I say that the transport exporting T, it requires RAN GTP means it requires the presence of RAN GTP. Why? Because RAN GTP facilitates the binding of exporting to the RNA molecule and it directs the complex to the cytoplasm. So if this is your RNA molecule and it has to go from the nucleus to the cytoplasm. So it directs the uh, movement of the RNA molecule from the nucleus to the cytoplasm. Now, then why not other options? Because RAN GAP. it promotes the hydrolysis of RAN GTP to RAN GAP. Okay, in the cytoplasm. So what happens in the cytoplasm? RAN GTP is converted into RAN GAP. So this hydrolysis which happens in the cytoplasm, this is supported with the help of RAN GAP. So RAN GAP, it doesn't help exporting function. It helps in the degradation of GTP into GDP. Okay, then I come to the next one, exportin 5. How why exportin 5 cannot be done? Because exportin 5 is specifically involved in the export of pre-mRNAs only, pre-miRNAs. So it is involved in the export of pre-miRNAs. So this cannot be the answer. Then RNA quantity inside cell nucleus, because it does not regulate the exporting activity directly. More specific answer is RAN GTP. So this is why the correct answer is RAN GTP. So I hope this is clear. Okay, somebody wants to join the call. Okay, now we come to the next question. Next question is FG nucleoporins are located in A. Nuclear membrane, B. Nucleolus, C. Nuclear pore complex, and D. It is cytoplasm. So where the FG nucleoporins are located? Nuclear membrane, nucleolus, nuclear pore complex, or in cytoplasm? This is your question. Right now it is 8:15. I will switch the screen after 2 minutes. That is 8:17. Whoever knows the correct answer can type it in the message box. Okay. Please try to answer some questions. Okay. One more minute left. Thirty seconds more. Okay, now I am switching the screen. Okay. Let me see what you have written. Nobody has answered anything. Why? You don't know the answer? This is something that we have already studied last time, no? So, the correct answer is nuclear pore complex. So, FG nuclear porins, we have already studied about this now. This helps in the export. So, these FG nucleoproteins are, FG stands for what? Phenylalanine and glycine. FG stands for phenylalanine and glycine. And these nucleoproteins are located in the nuclear pore complex, which helps in the import and export. We have already studied this. So basically, these are FG nucleoproteins are nothing but they are specialized proteins that line the nuclear pore complex, and it forms a selective barrier for the nucleocytoplasmic transport. Always remember that these Fg proteins, nucleoporins, Fg, it forms a selective barrier. So this does not allow the transport of everything, anything and everything. Just like we say that we have a semi-permeable membrane. It will allow, it is permeable only to a few things and not to everything. So likewise, this nucleoproteins, Fg nucleoproteins, which are made up of phenylalanine and glycine, they are specific and it acts as a selective barrier, and they contain the Fg repeats, that means phenylalanine glycine repeats, which create a mesh-like structure that regulates the passage of molecules between the nucleus and the cytoplasm. So this phenylalanine and your glycine, it is acting like a mesh-like structure, and this mesh-like structure, it actually uh, uh, restricts the passage of anything and everything between the nucleus and the cytoplasm. So this is the answer. So the correct answer is nuclear pore complex. Okay, fine. Your next question is, this so you should know. Length of poly A on mRNA tail indicates the mRNA is undergoing decay, that mRNA is ready for translation, age of the mRNA, or the nuclear location. What does the length of poly A tail on mRNA tail indicates? Right now it is 8:20. I'll switch the screen after 2 minutes. Think and try to answer it, okay? Think. Today also we have discussed this point. One minute more. Try to answer the question in the message box. Okay, now I'm switching the screen. Let us see if you have answered anything or not. Hmm? Nobody has answered anything? You don't know the answer of this question as well. Length of poly A tail indicates what? The age of the mRNA. We have already discussed now that if the poly A tail has, this question was there last time also. If the poly A tail is If you have a greater poly A tail, that means the mRNA is new, because the exonuclease activity has not acted that much on mRNA. If the length of the poly A tail is not that much long, that means the mRNA is not new, because the exonuclease activity has already acted on the mRNA. So length of the poly A tail indicates the age of the mRNA. Correct answer is age of the mRNA. Okay. Next question is a disease that is the result of defective mRNA export is osteomalacia, osteoporosis, osteomyelitis, or osteogenesis imperfecta. Right now it is 8:24. I will switch the screen after 2 minutes. This question also we have discussed. This question was there last time also. You should be able to answer this question. Okay, I'm switching the screen after 2 minutes. One more minute left. Okay. 30 seconds more. Okay, now I'm switching the screen. Nobody has answered even now. Disappointing. Anyways, the correct answer is osteogenesis imperfecta. We have studied it in the last class, that if there is the export process, if there is any defect in the export process, an aberrant export starts happening. Aberrant export starts happening, and because of that aberrant export, this osteogenesis imperfecta disease is caused. Okay. Next question. RNA undergoes decay because it lacks the coding sequence, it has stop codon, it lacks the essential stability features, or it has intron. What is the correct answer? RNA undergoes decay. Why? It lacks coding sequence, it has stop codons, it lacks the essential stability structures, or it has introns. Right now it is 8:27. I'll switch the screen at 8:29. Try to answer it. Hmm. One more minute left. Think. Okay, now I'm switching the screen. Okay, fine. So I already showed the answer, but let us see whether no. So what is the question? RNA undergoes decay because it lacks coding sequence, it has stop codon, it lacks the essential stability features, or it has introns. So, what is the correct answer? The correct answer is that it lacks the essential stability features. Why? Because the essential stability features, when we talk about mRNA, the 5' end has the 5' cap, 7 methyl guanosine cap, and at the 3' end, you have the poly A tail. These are the essential stability features which are present in mRNA, which prevents the mRNA from undergoing any kind of decay. And if the mRNA does not have these features, the essential stability features, that is 7-methyl guanosine capping at the 5' end and 3' poly A tailing at the 3' end, then that particular mRNA is marked for decay. So the correct answer is it lacks the essential stability features. Okay. Now the next question is, miRNA maturation is dependent on DROSHA, DGCR8, dicer or RISC. On what mRNA का maturation depends on? Right now it is 8:31. I will switch the screen after 2 minutes. Think and try to answer the questions. One more minute left. Okay, now I'm switching the screen. So the correct answer, let us see if you have tried to answer anything. No. Okay, fine. So the correct answer for this is dicer. So we have already discussed that the, the maturation of the mRNA, it happens with the help of dicer, and basically what happens, then dicer acts in the cytoplasm. Okay. So it acts in the cytoplasm. So when mRNA goes from the nucleus to the cytoplasm, it is acted upon dicer, and when it is acted upon by dicer, it gets uh, 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 it forms two strands, the template strand and the passenger strand, and then one of the strand goes to the risk complex, and then it performs its function. So dicer acts in the cytoplasm, drosha acts in the nucleus. This is what is important. Next question is, nucleoporins are important in RNA transport export. True or false? Nucleoporins are important in RNA export. I'm giving you 2 minutes. After 2 minutes, I will switch the screen. Okay. 1 more minute left. Okay, so I'm switching the screen now. Okay. The correct answer is true. Why? Because nucleoporins are basically the selective barriers which helps in the transport of the RNA from to and from the nucleus. Okay. So the correct answer is true. Okay. Now we come to the next question. Now we'll try to answer them quickly because I don't think that there is any need of giving time to you. Different RNA species follow different pathways of RNA export. True or false? Think. I will switch at 8:38. Think. Okay. So the correct answer is that yes, RNA species follow different pathways for RNA export. There are different RNA species: mRNA, tRNA, rRNA, SNORNA, snRNA. So all these different RNA species have a different pathway for RNA export. So the correct answer is true. Then second is exosomes degrade RNA from one end. True or false? I will switch the screen in. I'm giving you 2 minutes to think of this question. exosomes degrade the RNA from one end. This though you should at least answer. This we have discussed so much in detail, no? Right now we are discussing about exosomes only, nothing else we are discussing. Exosomes degrade RNA from one end. True or false? One more minute left. Okay, so the correct answer is that yes, true. Exosomes, they indeed degrade the RNA from one end. So they degrade the RNA from the 3' end. We have seen that exosomes, they show the 3' to 5' exonuclease activity. Okay. Since they show the 3' to 5' exonuclease activity, which means that they degrade the RNA from one end. Okay. Now, the last question is, deadenylation do not favor RNA stability, true or false? 2 minutes. Right now it is 8:41. I will switch the screen. We will like in 8:43. Okay, now I am telling the answer. Okay. So de-adenylation do not favor RNA stability. True or false? It is actually true. Because de-adenylation means it will remove the poly A tail from the 3' end. Okay, we have in the starting only we have studied about the de-adenylation dependent pathway. So when we talk about de-adenylation, this means removal of the poly A tail. Okay, so when there is removal of the poly A tail, what will happen? The 3' end would be exposed. And when the 3' end is exposed, then what happens? It is more prone to RNA decay. So with this, we have completed all the questions. Let us very quickly revise what we have studied today. Okay. So we started with the three different pathways that are required for the mRNA decay. The first is the de-adenylation dependent mRNA decay, de-adenylation independent mRNA decay, and endoribonuclease mediated mRNA decay. In the de-adenylation dependent, what happens? That first the de-adenylation enzymes, that is CCR4 or PARN, comes. It removes the poly A tail, and then the mRNA can either undergo 5' to 3' decay, or it can undergo 3' to 5' decay. If it undergoes 5' to 3' decay, then what happened? XRN1 will come. Sorry. XRN will come and it will chop from the 5' end. If it undergoes from the 3' decay, then exosomes come and chop from the 3' end. Then independent means that there is no need of removal of the polyatides. Directly the decapping enzyme will come and it will remove it will chop the RNA from the 5' end. Endoribonuclease mediated decay means some endoribonuclease will come. It will chop the RNA from between. Now there is an exposed 3' end and there is an exposed 5' end. In the exposed 3' end, exosomes will come and bind, it will chop it off. In the exposed 5' end, XRN will come and it will chop it off. Then we have studied about the P bodies. What are P bodies? P bodies are the translationally repressed mRNAs and proteins which are related to mRNA decay. So in normal cells, what happened? LSM1 and XRN1 are not together. But in P bodies, in the stress condition, when mRNA is marked for decay, then LSM1 and XRN1 comes together and it forms such yellow structures. Okay. Then next is we have studied about the exosomes. What are exosomes? What are the exosome structures? Important point to notice that RNA degradation complex, these RNA degradation complexes always targets from 3' to 5' end. That means exosomes always function from 3' to 5' end. Then how the exosomes regulate various machinery. Then what is mRNA surveillance? That means quality check. So what it means that it will detect and it will degrade the mRNA, aberrant mRNA. So aberrant mRNA are slow. They do not have a proper 5' capping. They do not have a proper 3' tailing. They have malformed mRNPs and they have slow polyadenylation. So these are the characteristics of aberrant mRNAs. Then for in this mRNA surveillance in the cytosol, there are three major pathways: nonsense mediated mRNA decay, non-stop mRNA decay, and no-go mRNA decay. So when we talk about the nonsense mediated, what happens? That premature stop codon is there, which marks the mRNA for degradation. In non-stop decay, what happens? There is either no stop codon or stop codon is there, but ribosome is unable to stop it. Then that particular mRNA is also marked for degradation. No-go RNA decay means that some kind of secondary structure forms in between the RNA, and because of this formed secondary structure, uh, there is a halt or stall in the translation process, and then particular mRNA is marked for degradation. Then we have studied, there are various examples of autoregulation of RNAs, such as autoregulation of NAB2, that is the poly A binding protein, degradation of meiotic transcripts in vegetative cells, exosomes and gene regulations, and all these things. Then we were studying about the non-coding RNA. What are non-coding RNAs? Non-coding RNAs are the functional RNA molecules, or not mRNA, functional RNA molecules, but they are not translated into any protein. Those are non-coding RNAs. And there are two main classes of non-coding RNAs. First is the housekeeping non-coding RNA, and second one are the regulatory non-coding RNAs. The housekeeping non-coding RNAs are the RNAs which are constitutively expressed. All the time they are expressed and they are required for the normal functioning and the viability of the cell. Whereas the regulatory RNA, they are not expressed constitutively, but they are expressed in certain stages of organism development. Okay. And these regulatory RNAs, it can affect the expression of other genes on the level of transcription also, and they can affect the functioning of other genes on the level of translation also. So these are the things that we have majorly studied. Okay, so I hope the whatever we have studied, at least something that you could understand from this. And so this marks the end of today's class. We will meet again next Tuesday at the same time, that is 7 to 9. Okay, so if you have any queries, you can ask me. Does anybody has any queries? No, no queries. Okay, so we'll leave the class today. Just a minute. Okay, then you can leave the class now. Bye bye. We'll meet in the next week's class. Okay.